Okay?
I would like to thank you again for being part of this discipline. So, Professor Salini Cola is from the Albert Einstein College of Medicine, that's in the state of New York, in the US. He's spending a month with us. Who between you were in his talk that happened here on Monday? Well, some. So for those that were not here, he's a colleague of mine in neuroscience. He's specialized in the studies of the neuroscience of a part of the brain that's named the nucleus accumbens, for the students of biology and in the biomedicine that are not familiar with this. We'll see about it in more details later on in the discipline. And this is a part of the brain related to reward, related to drug abuse, and maybe with selections of proper behaviors. And he used tools to study this part of the brain that are mostly related to measuring activity of neurons in the brain. So there are several ways of doing it, and this is the main subject of his talk today. So thank you very much for being here.
My pleasure.
Your students.
Okay. Thank you, Claudio. So like on Monday, I'm going to apologize again because I don't speak Portuguese. I would like to invite all of you to interrupt me if you don't understand something. I'll try to go slowly so that everybody understands, but feel free to just speak up and raise your hand, whatever, say something, and ask me to repeat, and I'm happy to do that. So my lecture today is on methods for measuring neuronal activity in vivo, so in animals, in whole animal preparations. So I realized when I made this outline that I didn't have a slide explaining what we mean by neural activity. So neural activity is really the transmission of information from one neuron to the next, is the best way to describe it. And that's done usually via synaptic transmission, but of course also via propagation of action potentials from one part of the neuron to the next. And I'll summarize that and go over it a little bit in the next slide, or in a couple of slides. But first, I want to give you an outline of this presentation. I'm going to talk about first why we do this, what is the reason why neuroscientists are interested in measuring neuronal activity in whole animal preparations. And I'm going to discuss really two main methods for doing that: electrophysiological methods, as well as calcium imaging methods, which have recently been developed to do this. So I'll talk about exactly how these work and some of the advantages and disadvantages, and then just very briefly at the end, I'm going to remind you that there are some neurochemical methods, but I'm not going to go into a lot of detail about them. So first, why do we measure neuronal activity in vivo? Well, really because as neuroscientists, we're interested in how neurons encode information. This is really the primary objective of much of neuroscience, is to figure out how information is stored and represented in the brain and how it's communicated from one part of the brain to the next. So that's really why we do it. We also want to understand how neurons are connected in circuits. Of course, the brain is very, very complex. Individual neurons can make hundreds, thousands, millions of connections with other neurons. And we want to understand how these are connected. We know literally a tiny fraction of this this kind of connection diagram of the brain. And it's through this kind of measuring of activity, how one neuron influences another, that we can start to put this together and understand in more detail. We want to understand how synaptic connections change with plasticity as animals and people learn about the environment. This is due to a process of synaptic plasticity, and in order to understand how information is represented and transmitted, we have to understand, we have to again measure in the animal as it's performing tasks and learning, we have to measure this activity and relate it to learning. And of course, there are the classic questions in neuroscience, questions like, how does the brain do these things, like sensation and perception and control movement and produce decisions and produce cognition? All of these are questions that we can't answer without measuring activity. And we can, of course, there are ways of measuring activity in vitro, such as in splice preparations, that can tell us a lot about how the nervous system works. But there's really no substitute for doing this in vivo, in whole animal preparations where the circuit is intact, and preferably in behaving animals, so that we can really understand how these circuits, neural circuits, work to do these things. So before I talk about the physiological and calcium imaging methods, let me just first summarize how it is that, that neurons are connected. So this is two stereotypical neurons, one connected with the other with a synapse. And you can see here in the presynaptic neurons, I'm going to try to use this instead of that big stick and see if it works. No, it doesn't work. Okay. So I have to use the big stick. Okay, so in the presynaptic neuron, of course, it has a cell body, it has dendrites, and it has an axon. The dendrites gather information via synapses, and that information is gathered is represented as a membrane potential, so as a voltage across the membrane. And if this potential becomes depolarized enough, so normally it's hyperpolarized at minus 80 volts or something, millivolts or something like that. If it becomes depolarized above a certain threshold, it fires an action potential, which is a regenerative change in voltage that travels down the axon until it reaches the terminals. And in the terminals, an action potential will cause influx of calcium, which will trigger fusion of the synaptic vesicles to the membrane, and therefore release of neurotransmitter into the synaptic cleft. The postsynaptic neuron will have receptors. The neurotransmitter will bind to the receptor. This cell will then either be slightly hyperpolarized or depolarized, and through integration of all these small synaptic potentials, this cell will either reach threshold or will go away, be more hyperpolarized from threshold, and essentially that's how it decides itself whether it's going to fire an action potential. So this is the basics of how neurons communicate with each other. There are, of course, many extensions and exceptions and so on, but the important point for measuring neural activity is that there are a couple of changes in potential that we can pick up using electrodes placed in different parts of, either inside of cells or outside of cells. The largest change in potential is an action potential. This is on the order of 0.1 to 0.2 millivolts, so it's quite, volts. So it can be something like 100 millivolts or so in amplitude. So it's actually quite large. It's possible to detect this with the appropriate electronics with electrodes even outside of the neuron. We can also detect synaptic potentials, which highlighting here. Synaptic potentials are generated when ionotropic receptors, such as glutamate receptors or GABA receptors, are activated by neurotransmitters, and that causes a depolarization that's considerably slower than an action potential. It's not regenerative, but it's a potential that can also be measured using both intracellular and extracellular electrodes. And finally, there's the issue of calcium. Depolarization of the neuron causes influx of calcium both in the dendrites and in the cell body, and also in the synaptic, in the presynaptic terminal. So all this calcium can be measured using modern techniques for calcium imaging, which we'll talk about in the second part of the talk. But first, I'm going to talk about electrophysiological methods. So these fall into two categories. So electrophysiology basically means we're going to use an electrode to measure these potentials. And the two categories are intracellular and extracellular. So intracellular recording just means that the electrode is inside the neuron, and extracellular just means, of course, that the electrode is outside the neuron. In all cases, whenever I talk about electrophysiology in vivo, in behaving animals, in awake animals or anesthetized animals, any of these preparations, there's always going to be a ground electrode somewhere. The ground electrode is just a low impedance electrode that's for return of the current. You have to complete a circuit in order to measure it, and that's located somewhere usually quite distant from the electrode we're interested in. Because it's low impedance, it's not going to detect on its own any kind of activity that's happening around it, and so we're going to sort of ignore it and pretend it doesn't exist. But from a physics perspective, we always have to have a ground electrode in order to measure a change in potential. Okay, so intracellular methods. There are two main kinds of intracellular methods. They really depend mostly on the kind of electrode that's used. These are both used glass electrodes. One of them uses what's called sharp electrodes. Sharp means that the tip here is about 1 micron in diameter, and these glass electrodes are filled with a saline solution that matches what's inside the cell. And so the way that this works is that these penetrate the neuron because they're sharp, as you might imagine, whereas the whole cell electrodes are considerably larger. There's something like 3 or 4 microns in diameter, and they work differently. You form a seal with the membrane, which I'll describe in a minute. But first let me talk about sharp electrodes. So like whole cell electrodes, they allow the membrane potential to be recorded. So the purpose of this is to measure the potential across the membrane. So you have the electrode inside, here's the membrane, and you have the ground electrode somewhere way out here, and so you can measure the potential across the membrane. That's the purpose of both of these kinds of recording. So of course the memory potential is going to represent, there's going to be both action potentials, and there's going to be synaptic potentials. And there's going to be potentials arising from other things that might happen to the cell, such as if you depolarize the cell enough, certain cells will have calcium spikes, for instance, which are just another form of action potential, but they're limited to calcium channels. So any kind of thing that changes the potential of the membrane, you can pick up with this kind of electrode, either of these kinds of electrodes. Sharp electrodes allow you to record from relatively deep structures. You can keep on moving the electrode down through the tissue, and even if you get some junk at the tip of the electrode, it doesn't matter. You can just still penetrate another neuron. That's one reason why they've been used quite extensively, especially in the early history of neuroscience. It tends to be quite difficult to hold cells, because if the brain moves a little bit in relation to the electrode, then you no longer have your electrode inside the cell and you don't see anything. So very often, unless your preparation is extremely stable, these kinds of recordings last for only a few minutes. And for that reason, this is really only used in either anesthetized or head-fixed preparations. Typically only anesthetized animals, but there are ways now of training animals, such as mice, to allow themselves to have their heads fixed in place so they can't move their heads, and then so you can do these kinds of experiments in awake animals. It's not done very often with sharp electrode recording, but it is done. And so this technique is, I would consider it extremely difficult, partly because it's so hard to hold neurons, because the preparation has to be so stable, but also because the shape of the electrode is very, very important, and it's almost an art and not a science to actually figure out how to get the electrode inside the cell, and so that makes this something that is very hard to do. Whole cell recording, on the other hand, is slightly easier. Both of these techniques are easier to do in vitro, such as in the slice preparation, but they are done in vivo as well. Whole cell recording also allows you to measure the memory potential, but it actually gives you better control of the potential. So the way this works is that you bring the electrode up to the membrane, and this forms a seal. You apply slight suction to this to sort of suck the membrane up into the electrode, and then break the seal with a sharp suction, okay? And so now you have access between the electrolyte solution in here and the cell, and a gigaohm seal with the membrane, which means you have a relatively high impedance connection between the extracellular space and this whole system of the electrode and the neuron. Okay. So because of the surface area of this connection here is relatively large, it's very easy. It's a low impedance connection between the cell and the electrode, and so electrolyte can easily sort of move back and forth, either via current or via mass flow, either one. And that makes it very easy to control the membrane potential as well as to record it. So you can use techniques such as voltage clamp, which again are typically done in vitro preparations such as slice, but can also be done in vivo. This is typically only done in shallow structures, such as in the cortex, because if you get a bunch of junk on the tip of the electrode, as you lower it through the brain, it will not make a good seal with the next neuron that it encounters, and you can forget about getting a nice whole cell recording. So this is usually only done in the cortex. It's a little bit easier to hold cells. It requires anesthetized or head-fixed preparations. It can't be done in freely moving animals because, again, if the electrode moves in relation to the electrode, you'll lose the recording. This is also, I would consider this very, very difficult. And this is one reason the the difficulty of this and the low yield of recordings per sort of day that you try to do an experiment is one reason why people have developed other methods for trying to record from neurons in vivo that are a bit more efficient, or actually a lot more efficient. Before we move on to extracellular methods, there's one other method we should talk about that's kind of a hybrid between these two, and this is called juxtacellular or cell-attached recording. So you use the same kind of electrode as your whole cell, and you try to make a seal onto the cell. It doesn't have to be a very good seal, you just basically have to have the electrode up close to the cell, and you don't try to break into the cell by rupturing the membrane. You just essentially have the electrode very close to the cell. And what this allows you to do is to measure potentials that are relatively large. So you can pick up action potentials using this method, but you can't pick up smaller changes in potential like most synaptic potentials. Unlike some of the techniques that I'm going to talk about in the next few slides, you can be very confident that you're recording from only one neuron, and that's it, because you're very close to that neuron. Somewhat surprisingly, this method actually allows you to label the neuron that you're recording from. You can do this with whole cell recording, but with this method you can do this as well. So you can fill the electrode with dye, and of course it would seem like because you don't have a connection between the electrode and the neuron, that the dye can't get into the neuron. But in fact, most dyes are charged. So if you pass a current through the electrode, some of that current will be carried by the dye, and if you use a high enough current, it will actually disrupt the membrane and get into this cell. And so what you can then do is if you load the cell with enough dye, you can remove the electrode, kill the animal, and cut sections, and try to figure out where the neuron is. Once you've found it, you can stain it for things using antibodies to determine what kind of neuron it is, what kind of neurotransmitter it has, what kind of receptors it has, what kind of molecular properties it has, and so on. So as you can imagine, this is quite time-consuming because literally it's almost like it's worse than finding a needle in a haystack to try to find this one dyed neuron after the fact, one neuron in the whole entire brain. But it is possible and people do it, and it can be quite informative if you do the right kind of experiment here to record from this neuron and ask how it responds in various different conditions. And this is usually only done in anesthetized preparations or in a wake head fixed. And as I just mentioned, it's very, very time consuming. So those are intracellular approaches. I'm going to talk now about extracellular approaches. So where the electrode is outside of the neuron, you could record these kinds of potentials. We call them LFPs, EEGs, EEGs, electroencephalogram, and a couple of other approaches that are like this. And then we'll talk about single unit recording. Those are all extracellular methods. So how does this work? Again, we have an electrode. It could be in the brain, but outside of a neuron. Again, there's going to be a ground electrode somewhere. This is one way of doing it. Another way of doing it is actually not to have the electrodes inside the brain, but to have the electrodes on top of the brain. These electrodes are actually on top of the dura. And this is called ECOG, or electrocorticogram, which is often done in primates. So there's actually a whole bunch of electrodes here on top of the dura. And so this is very similar to EEG, but somewhat more invasive. EEG is electroencephalogram. You can do this, of course, completely non-invasively in people, or in animals, if you like, where the electrodes are actually on the scalp. And as we go from more invasive to less invasive, the spatial precision of the recording gets worse and worse, right? So over here, you can record from potentials that arise in the vicinity of the electrode. And here you can record from potentials that arise from the underlying cortex. But here you have really no idea where the potentials are coming from, or maybe some idea if you talk to the EEG experts. But really, an electrode over here can record from a potential that arises all the way on the other side of the brain, and you'd never really know. And the reasons for that are because of the physics of this kind of recording, which I'm going to describe in the next few slides. So all of this kind of recording of LFPs, LFP stands for local field potential, and I'll describe why that's called that in a minute. All these kinds of methods are essentially trying to pick up a voltage that's the result of a current that arises from actually more than one neuron. But for this example, let's imagine we have one neuron, and there's a whole bunch of extracellular fluid, cerebrospinal fluid, and we have an electrode over here. So this neuron generates a current because it's either fires an action potential or has a synaptic potential. And we have an electrode here that's going to pick up a voltage as a result of that current. Well, if we're going to have a current source and we're going to have an electrode that picks up a voltage, well, what is the voltage going to be? So have you heard of Ohm's law? Is that familiar to everybody? Ohm's law? Is that translated into Portuguese? Is it the same? Yeah, okay. V equals IR. Voltage equals the current times the resistance, right? Well, that's what's going to determine what the voltage is. Okay, so let's go over this, this equation one by one. Okay, so VR is the voltage at the electrode. I0 is the current generated by the current source. And, well, what's all this stuff over here? This doesn't look like R. R minus R0 is the distance from the electrode to the current source. So R just basically refers to radius because this has to do with the fact that this is kind of like in a sphere. So R minus R0 is this distance from the electrode to the neuron. And sigma over here is the opposite of resistivity. It's the conductivity of the medium, okay? The conductivity is how conductive it is per unit of distance, okay? So the greater the distance between the current source and the electrode, the greater the resistance is, right? The lower the conductance is. Because there's just a lot, there's just a lot of space and a lot of resistance, and there's more resistance with distance. So to give you an example of this, from a more sort of hardware perspective, if you think about a circuit that you've all, at some point, because I'm sure you've taken physics, thought about, you might have a circuit with, say, a battery and a motor, and then there's wires connecting those, right? And we think of those wires as having no resistance at all. But in fact, that's not true. They have a tiny bit of resistance. And if you just have a short distance of wire, say a few centimeters, or even a meter or so, that resistance is effectively zero, because it's designed to be that way, because metal is very, very conductive. But if you take 10 kilometers of wire and try to put that in your circuit, the resistance now adds up because there's the resistivity, or the essentially sums across all this distance that you're trying to push the current through. So if you use 10 kilometers of wire versus 10 centimeters, that resistance is now very appreciable. It's going to contribute to what the voltage you actually measure and so on. If you apply a certain voltage, it's going to be the current is going to be influenced by that resistance because of Ohm's law. Okay, so the principle is no different here. So V equals IR, voltage equals the current divided by the conductivity, and the conductivity is a function of a constant, which is sigma, and the distance. Okay, is this clear to everybody? Nothing more than Ohm's law. So, of course, in most cases we have more than one neuron. Our brains have a few more than that. So you have, let's say, 3 or 4 neurons, and you have an electrode here. What is the voltage going to look like? Well, it's going to be the sum of all of the voltage change is going to be the sum of all these sort of Ohm's law currents that are produced by all these different neurons. So that's just this over here. So the voltage is the sum of the individual currents, right? Divided by the distance and multiplied by this constant, which has to do with the conductivity. Okay. So it turns out that this equation is actually incorrect. This was the first equation that people came up with to try to describe this. I'm not going to describe why it's incorrect, but it has to do with the fact that it makes this assumption that the conductivity is actually constant across distance, and that's false. The reason why it's false is because if there's an extra neuron or axon or dendrite or myelin or whatever in the way, that's actually going to change the conductivity of the interval between the electrode and the neuron. And so that's why this equation doesn't really work in practice. But as an idea of sort of a summary of how it works, or to give you an intuitive idea of how it works, this is it. The other thing that this thing doesn't capture, well, it captures this, that as the as the distance increases, the measured voltage for a given current decreases, right? That makes sense, right? Because we're dividing by the distance, right? But what actually happens is that this decrease is greater for faster frequency changes in current, okay? So, for instance, and this is called, I should just give it a name because it's easy to refer to it, it's called frequency filtering. So if you have a very fast change in current, then this equation is actually wrong, and this voltage will actually be much more attenuated than for a slower change in voltage. And this has a practical consequence, which has to do with the fact that action potentials are very high frequency changes in current. They're all over within a millisecond or two. Whereas synaptic potentials are very slow changes in current. They're over within 100 to 200 milliseconds. They're 2 orders of magnitude longer, right, or slower in frequency than action potentials. So let's think about what that means. The implications of frequency filtering are that for action potentials, which are high frequency changes, the electrode has to be close to the neuron in order to detect the action potential. If the electrode is too far away, you're not going to detect the action potential. Okay? So usually with one electrode that's relatively high impedance, you can measure action potentials for maybe 1 or 2 or maybe up to 5 or 6 neurons, but typically not more than that. And so if the neurons are too far away, you won't pick up the action potentials, but it's actually the opposite for synaptic potentials. Okay? So synaptic potentials are much lower in frequency, so they're about 100 milliseconds or 200 milliseconds long or something like that. So what that means is that an electrode placed in the brain or even on top of the brain, such as an EEG electrode, is going to pick up the synaptic potential signal from many, many, many different synapses over a large volume of tissue. Okay? And in fact, it's impossible to stick an extracellular electrode just into the brain somewhere and expect it to pick up the potential change from just one or a couple of synapses. It's such that you're always going to pick up the potential change from many synapses at once. Okay? That's what I just said. Okay, so we can record action potentials from just a few neurons. We can record synaptic potentials from a lot. It turns out that what is commonly called, this is a jargon term in electrophysiology, local field potentials, or LFPs. These are just slower changes in potential than action potentials. For a long time, it wasn't quite clear what these came from, but it turns out I think everybody agrees now that these are mostly from synaptic potentials. So if you hear the term LFP, or local field potential, it refers basically to an electrode that's measuring synaptic potentials, and it's always the sum of many synaptic potentials from a fairly large volume of tissue, not from just a couple of synapses. Okay? And that is especially the case for these kinds of less invasive methods where the electrodes are quite far away from the brain, and therefore they're very low impedance electrodes, which means that they're picking up the synaptic potentials from large volumes of tissue. And as I mentioned, for EEG especially, you can pick up potential, say over here, in the temporal cortex that arise from the occipital cortex, and you have no idea whether that's the case or not, where exactly they're coming from. There's no way of localizing it.
Okay. I have a question. Well, for everybody, maybe you could definition of impedance.
I'm sorry.
For these, this is a real doubt I have.
Okay.
So why high impedance electrodes are better for spikes and low impedance for local field potential?
So you can measure local field potentials with high impedance electrodes. It's just that they'll come from just a local area, right? And synapses and synapses typically don't fire, so synaptic potentials are typically not very synchronized, so they tend to cancel each other out a lot. So in order to see anything, it's like, imagine that you have 10 synapses that are somewhat synchronized with each other. There's going to be a lot of noise in that because you're averaging across 10, and sometimes it's going to be large, sometimes it's going to be small. Let's imagine that you now have 1,000 synapses, and they're, say, 10% synchronized. Well, across 1,000 synapses, the noise kind of cancels out, and so the signal where all sort of changes at once, you can sort of pull that out, right? So with the lower impedance electrodes, you're collecting data from more synapses, and so you can pick up a bigger change that's sort of more synchronized. At least I think that's how it works, right? So, and you asked for a definition of impedance. There is a difference between impedance and resistance, but I've forgotten what it is. It's basically, the way I think about it is it's resistance, right? It's just, my brain is geared towards Ohm's law, V equals IR. That's what it is. Yeah. Any other questions? Okay. All right, so how do you pick up spikes in LFP? So Claudio alluded to using low impedance electrodes to pick up spikes. In fact, you can use low impedance to pick up LFPs. In fact, you can use low impedance electrodes to pick up spikes, and you can use high impedance electrodes to pick up LFPs. It's just that the efficiency with which you do it differs. And in fact, there's many kinds of electrodes that will do perfectly fine picking up either kind of signal. And that's I'm going to show that in this example here. How is it that we separate out LFPs from spikes? So this is actually illustrated very nicely in this experiment that was done in this paper here, I think the idea was to actually illustrate this. It wasn't to actually test any hypothesis, but it illustrates this very nicely. So what they did in this experiment is they have an intracellular electrode. So they're recording from a neuron using the intracellular. They have an extracellular electrode, so they can measure this, what happens both intracellularly and extracellularly at the same time, right? So that allows us to compare intra and extracellular. And so this is how it works. This is the intracellular recording. So this diagram, this graph here is just the voltage across the membrane over time, okay? And what they've done here is they've actually, because they can control the voltage, they've depolarized it so it's above threshold. And you can see here that it's firing some action potentials, okay? So over time, this is 40 milliseconds, this bar. This is 80 millivolts. This neuron fires a few action potentials, okay? Well, now they also have an extracellular electrode. So they can ask, what does that look like? So this is what the trace looks like, okay? So notice, I mean, I don't know, can any of you find the action potentials in this trace? I mean, it's like hard to see, right? So I can find maybe, oops, so this actually used to work, this little pen thing, but it's, I see that's, I have to press this thing. There we go. So there's a spike. And there's a spike, and I don't know, maybe here's a spike. The one that corresponds with this seems really tiny. You see it there, right? The one that corresponds with this one here, maybe that one, and this one. So you can see they're extremely tiny, right? So this seems like a real problem. How can anybody pull out action potentials from this ridiculously noisy signal, right? So the problem really is that these things are so brief, yet the majority of the signal is this kind of slow up and down kind of stuff that lasts, you know, on the order of 40 milliseconds or something. Well, we know action potentials are only a millisecond or so. So this little tiny stuff is getting lost in the noise. So what do we do about that? And so the answer is filtering. So you can use either a hardware filter, or you can do this to the recording. If you digitize this, you can use, in software you can apply a filter as well. And there are essentially two kinds of filter that do two different things. One is a high-pass filter and the other is a low-pass filter, okay? Pass means what kind of information passes. High-pass means that the filter allows high-frequency changes in voltage to pass, and it cuts out the low-frequency changes. Another name for a high-pass filter is a low-cut filter, okay? So in this case, they applied a high-pass filter. So that means that it threw out all these slow changes in voltage here and allowed only the high frequency changes in voltage through, okay? So the scale here is a little bit different. Up here, this bar is 0.3 millivolts. And for this trace here, the bar is 0.04 millivolts. So it's an order of magnitude sort of more magnified, okay? And you can see here now that very nicely, first of all of this slow up and down crap here that is interfering with us detecting the spikes goes away, and it's a nice flat signal here, and also the action potentials are showing up very nicely superimposed on this very even, very flat kind of filtered signal, okay? So filtering is critical for electrophysiology. Without filtering, we couldn't do anything. Conversely, if we wanted to pick up the LFP, we would apply the opposite filter, right? Instead of a high-pass filter, we would apply a low-pass filter, which is, another word for that is a high-cut filter, which means we cut out the high-frequency stuff like these action potentials. And probably one reason why there's all this slow changes in voltage here is because there are synapses firing away somewhere nearby, and they're being integrated and summed by the electrode, and in fact, when they're slightly synchronous, you get these kind of slower changes. So if you apply, and they don't have an example of this in this paper, but if you apply a low-pass filter to this, then you'll see the LFPs emerge quite nicely without this kind of high-frequency stuff that comes from action potentials, okay? So why do we do this? What can we do with this kind of data? Often what's done is the filter data is aligned to behavioral events, right? So, for instance, if you are doing this in a behaving animal and you present the animal with a stimulus, or you ask the animal to do an action or to make a decision, you can align the data to that point, repeat the trial many times, and then, excuse me, if you see systematic differences, sort of before versus after you presented the stimulus and did the manipulation, you can draw conclusions about what the signal means. So typically if you have an LP or EEG electrode, you're asking about the activity summed across many synapses, and you're asking how all those synapses change. Of course, some are going to go up and some are going to go down, but the bulk of them might go in one direction or another. And so that's one limitation of this method, is that we don't really know what an individual synapse is doing, but we know what the whole bunch of synapses are doing. But we don't even necessarily know which synapses they are. But we get an idea of how one part of the brain might respond. And so for low spatial resolution recordings such as ECOG and EEG, there's a whole field of neuroscience devoted to measuring what they call event-related potentials, which basically do the same thing but with, as I just described, but they're integrating over very large volumes of brain tissue. And as I've mentioned now several times, it's sometimes very hard to determine where those signals arise from because the integration is across such a large volume of tissue. Another reason for looking at LFPs is for measuring oscillations. So there are sometimes the activity of the brain becomes synchronous and sometimes it becomes less synchronous. So one example is when you're asleep or under certain kind of forms of anesthesia, brain activity can become very synchronous. And this can easily be picked up with EEG electrodes or with LFP electrodes in the brain. And at other times, animals that are awake and behaving will have different oscillations at different frequencies in different parts of the brain in relation to different forms of activity, such as seeking behavior or exploratory behavior versus and paying attention versus not paying attention, and so on. And so this is also one way in which this data is interpreted. Okay, so let me finish the physiology section by describing single unit approaches. So there are several different kinds of electrodes that can be used, which I'm going to summarize here, and they have their advantages and disadvantages. And so the first kind that was used in mammals and in sort of higher organisms, in say Aplysia and the squid giant axon and so on, were these kinds of electrodes here. These are sharp metal electrodes. Sometimes they're glass electrodes. They're filled with a high concentration of salt solution. But typically it's metal electrodes with a tip diameter of about 1 micron. Okay? So I've drawn this to scale here. This is 1 micron. The diameter of this circle is 10 microns, and this one is 25 microns. And I picked these two because these are typical diameters of cell bodies of neurons in the brain. So cortical neurons have diameters of about 25 microns. And neurons in the striatum have diameters of about 10 microns. And this is a typical range. 25 microns is about as large as they get, and 10 microns is about as small as they get. And there's a few that are a little bit smaller, but not many. So these are high impedance electrodes. There are 1 to 3 megaohms. And I write here at 1 kilohertz because impedance is measured using a sine wave. And quite often people pick 1 kilohertz because action potentials are 1 millisecond, which means about a kilohertz. And so the impedance actually that you measure will be different depending on the frequency of the oscillation. That has to do with the property of metal electrodes that I don't fully understand, but it's there you are. The tip diameter is about 1 micron. Typically, tungsten, stainless steel, or certain metal alloys are used to make these electrodes, and then they're insulated right up until the tip. These kinds of electrodes are typically used only in acute experiments. What this high impedance allows you to do is to record from action potentials only from neurons that are very, very close to the electrode. If you have lower impedance electrodes, you can collect the signal from a slightly larger volume of tissue and pick up action potentials from more neurons, okay? So because of that, because your electrode has to be so close to the neuron that you're recording from, you have to position it very, very carefully. So you have to use a very high precision micro manipulator, usually with a stepping motor, to position this into place. And if, of course, the neuron moves in relation to the electrode because the animal moves or something like that, then you're going to lose the cell and not be able to record from it. So this is really only done in acute preparations with head fixed or anesthetized animals. I just said that. This gives you very, very good signal to noise ratio in the sense that because your electrode is so close to the neuron, you can control it with your micromanipulator. You'll have a very, very large action potential signal. and the noise will be very, very low. So, and typically there's only going to be one neuron, or maybe at most two if there happens to be another one very, very close by. And it'll have a different shaped waveform, so you can really easily, you can be very confident that your neuron is just one neuron that you're recording from, and that there aren't other neurons interfering with your signal. So there is a low yield of neurons per experiment with this, which is why this is not really used very much anymore, although there are some labs that still use this method. Another method is to use a lower impedance electrode. My lab, for instance, uses electrodes that are 50 microns in diameter. And so again, I mean, the contrast is huge between these sharp electrodes. So the impedance of these is about an order of magnitude less, okay? Which means that we're picking up a volume of tissue that's considerably larger than with the high impedance electrodes. I'll explain that in a minute. Other labs use diameters that are a little bit smaller, something on the order of 20 microns, but the impedance is still quite similar. Materials that have been used are stainless steel, this nickel chromium alloy called nichrome, or in my lab we actually use tungsten because it's very, very easy to use. It's nice and stiff. These are typically used only for chronic implants. And so we don't quite understand why, but I have some ideas why this doesn't work with acute preparation. So when you lower this large electrode through the tissue, and then stop, and then try to find a neuron, you won't find any. You have to wait about a day, sometimes 2 days, before you start to see signal on the electrode. Well, why is that? Well, I'm not sure, but I can make something up. And that is that because this electrode is so large, it's going to cause a lot of tissue damage compared to this very, very, very fine tip here. There's only 1 micron tip. This 50 micron big bulky thing is going to kill a lot of stuff. Okay? So there's going to be lots of neurons and other cells, glia, that are going to be releasing their internal contents, their cytosol into the extracellular space. There's a lot of potassium involved, there's calcium, there's all kinds of stuff. There's probably going to be an inflammatory response. And so the surrounding neurons are probably under some kind of stress. They might be very depolarized because of all the potassium floating around from all the dead neurons. And so they're not going to be very responsive. But if you wait a day, everything looks fine, and you can pick up neurons on the electrode. So typically the way that an experiment works is that you implant the electrodes, and you can either just leave them there and fix them in place relative to the skull, and then these will be very, very stable across many days. Or quite often people will put these electrodes into a microdrive, and so that you record from neurons in one day, you advance the electrodes, and come back the next day and record from a new population of electrodes. Okay? And often this is done with an array or a bundle of electrodes, so you can record from many neurons at once. Okay? So animals can be freely moving for whatever reason, probably because let me illustrate it this way. When you have this high impedance electrode, let's say that this is my electrode, right? And this is my neuron, right? It has to be right up close to the neuron to see anything. And if there's even a slight movement, you lose the cell, okay? But now let's imagine that we have this big giant, I mean, if this is 1 micron, what is 50 microns going to look like? Like this, right? And here's my cell. Well, if this moves a little bit, who cares, right? Because it's still going to be there's going to be a lot of conductive space right here where the electrode is, and if this moves a little bit, well, you're still going to pick up the signal, right? So this is actually, you can get very stable recording. Some people claim you can record from days and months. I think there are certain people who claim you can record for years at a time from the same neuron. I don't know if I believe that. But it's certainly very, very stable compared to these high impedance sharp electrodes. As I said, usually multiple electrodes. I already described that. And this has low signal to noise, and the reason for the low signal to noise is, it's kind of counterintuitive because the impedance is so low, you'd think that the signal to noise would actually be, it's a reminder to give this lecture. You would think that the signal to noise ratio would actually be worse, but in fact, but the reason why, it would actually be better because of the low impedance. The signal to noise ratio is actually worse than for high impedance electrodes because first of all, you don't really have control over where the electrode is in relation to the neuron because you don't use a precision microdrive. But also because of the low impedance, you're collecting signal from a larger volume of tissue, and all the action potentials that are generated a far distance away produce a little bit of sort of chop in the signal. The signal goes up and down. Not necessarily enough to reach threshold, but sometimes the neuron that you're recording from is just barely above that kind of noise. And this is one of the problems with using this kind of low impedance electrode. Okay, there's one more thing. There's a high yield of neurons per experiment, often because you can get more than one neuron, sometimes up to 5 or 10, or I think we even seen something like 12 or 15 neurons on one electrode, okay? And they'll have different action potential waveforms, which is how you separate them, and that's one reason why you can get a high yield of neurons per experiment. But that does raise a problem, right? Which is, how do you assign spikes to individual neurons, okay? So if you have an electrode here, like this neuron, this electrode, and you have a bunch of 25 micron diameter cell bodies here, and they're all generating action potentials, and you're measuring them all with this electrode, how do you know which one comes from which? And the answer roughly is that the shape of the action potential is going to be different for each neuron because it's governed by the distance of the electrode to the neuron, okay? So this is just an example recording that I made as a postdoc many, many years ago. And it just shows two action potential waveforms recorded from the same time on the same electrode. And you can see that they actually look very, very similar, except that the amplitude is really different of the sort of the upgoing phase and the downgoing phase of the action potential. So why is that? So that's actually also illustrated very nicely by this same paper that did the simultaneous intracellular and extracellular recordings. So this is what they did in that paper. They have an electrode, sort of set of 6 electrodes that they have placed up near a hippocampal pyramidal neuron, okay? And they're recording the spiking of this neuron. So one spike produces these 6 signals, okay? And notice that electrode number 1, which is fairly far away from the cell body, it's up here in the dendrites, doesn't produce much of a signal, but as you get to electrode number 6, which is right up close to the cell body, you see a fairly appreciable signal. Then they move these 6 electrodes down further, so you see now number 1 is still in the dendrites, but it's closer to the cell body, and number 6 is all the way down here near the where the axon is. And you can see here that the signal changes according to how far away the electrode is to the cell body, okay? And this is exactly the principle that's used if you're recording from multiple electrodes at once. Different neurons will be different distances away, and so therefore the shape of the signal, not really the shape so much as the amplitude of the different components will be different. But it's theoretically possible, and in fact it's actually extremely likely that quite often you will have 2 neurons that are exactly equidistant from the electrode, right? Well, what about that? How do you separate those apart? And there are ways of doing that I won't get into if you just have one electrode sticking here. Those ways are not very efficient, but this is the reason why people invented stereotrode and tetrode recording. And this is how this works, okay? So here's the cross-section of a tetrode. A tetrode is just 4 of these low impedance electrodes stuck together. So the electrodes are like this, but there's also insulation around them that I'm not drawing. So this whole thing is quite fat. These electrodes are typically not 50 microns or something like 15 or 20 microns, plus insulation, but they're all stuck together as sort of one unit. Well, what's the logic behind doing that? Well, it's because although the distance between 2 neurons and an electrode might be equidistant, like for instance those 2 neurons are equidistant from that one electrode, they're not going to be equidistant to all 4. So not for the here. Here they might be equidistant, but they're not equidistant between these two. For this electrode, right? So for these 2 electrodes, it looks like they are equidistant, but for these they're not. That's the logic behind having 2 or 4 electrodes stuck together in either a stereotrode or tetrode formation. If you record from these simultaneously, you might have one electrode where you can't tell 2 different neurons apart, but on another electrode, the waveforms will look different enough that you can actually separate them. So using tetrodes and stereotrodes has become quite popular. The theory is really quite much similar to the low impedance electrodes that I already discussed. Same kinds of materials, better unit isolation. The disadvantages are that these electrodes are time consuming to make compared to just, in our lab we use just single electrodes, 50 micron tungsten. We literally use a pair of scissors to make them, we cut them. That's the end of the fabrication process, at least at the tip end. And so you can imagine how rapid it is for us to make an array of electrodes. Whereas if you're going to make tetrodes, there's a whole process you have to go through to put 4 electrodes together. The best way of using tetrodes to maximize yield is to drive them individually. And this is, but if you use, and typically if you're going to have, say, an array of 10 tetrodes, you have 40 individual electrodes. You're going to drive 10 tetrodes individually. That requires a very large drive. Some labs do this, but it's usually so heavy that a rat can't hold it, certainly, so you have to use a counterweight to keep it above the rat's head. So you can imagine what a mess this is for an awake behaving animal. If you use just individual electrodes and you drive them all at once, and then you can with stereotrodes or tetratrodes, you can just drive them all at once. So it's not a requirement to drive them individually, but there are some labs that do that because they want a very high yield of neurons per experiment, and I'll explain why that is in a minute. If you just drive all the neurons at once, it's considerably easier than if you drive them individually, which again you can do with tetratrodes. So one last thing before we go on is there's another kind of method of extracellular recording which involves using a silicon multi-contact probe. And the idea here is that this is nothing more than a printed circuit board, okay? A precision printed circuit board with the contacts separated by about, say, 25 microns, okay? So this is very much like having tetrodes, except there's many more than just 4 electrodes. There's a whole bunch in a row. So you can take this one here is 790 microns, right? So that's what, almost a millimeter. And so you can stick this down in the brain, and it's very easy to use. You just put it into the head, and you're done, right? And you can start recording. The disadvantage is that these are expensive. They're typically only used in non-human primates. I haven't seen many experiments where they've used this in rodents, although it has been done. And they're typically only used for acute experiments because I think it's harder to keep cells stable, although there are some people who report stable recordings acutely as well, so go figure. So this is another method that is commonly used, again, because it's so easy. Well, what do we do with this kind of data? So I could give, well, I'm not sure I really want to, but it would be possible to give 10 lectures on data analysis methods for this kind of data. But I don't want to do that. I'm going to give you one slide instead. And the basic idea is that the information that you can get from this kind of experiment has to do with the stimuli that essentially the events that you present to the animal. So you can do this in anesthetized animals, like for instance the Hubel and weasel experiments where they recorded from the visual system of cats. The animals were anesthetized, but they presented events to the animals. They gave the animal visual stimuli. They were different visual stimuli, and they asked how the neurons responded to those different stimuli. In behaving animals, one thing that we do is we give animals different stimuli, such as one cue that means reward, the other does not, and we ask how the neurons, we just align firing to those events, and we ask how the neurons respond differently. So for instance, we can make a raster plot where each tick mark here represents when the neuron fires. This is just time on the x-axis. And we align that to the cue onset and compare, by giving different kinds of cues, we can compare the firing and say that the neurons encode this kind of information. And by having the cues mean different things, we can try to discover what kinds of information the neurons encode. Okay, that's called an encoding approach. There's another approach called a decoding approach, where the idea is what you try to do is you record from a population of neurons, and you try to then predict the animal's behavior, okay? And one example of this that takes us to a complete extreme is the brain-machine interface. So I'm going to show you an example of this. This comes from Brazil's own Miguel Nicolelis, who loves to build brain-machine interfaces. And let's see if this actually works. I'm able to do this. Let's see. I got this to work a million times before, and now it refuses to. There we go.
You go over the slides.
Yeah, it's happening. There we go. So Miguel is now at Duke. So what you're seeing here is a monkey and a robot. He's driving the robot with his neurons, right? So what they have is a whole bunch of electrodes in, I forget which part of the cortex, I think it's the motor cortex. And they're interpreting those signals and controlling the robot with those signals, right? So the monkey has been trained to just basically think in a certain way and have a certain pattern of neural activity. One might mean go left, one pattern might mean go right, one might mean go straight. And so the animal can navigate in this field, and if he goes to the right location, they give him a bowl with grapes for him to eat, okay? So this is called a decoding approach because they are taking the neural information and they're predicting the animal's behavior. In fact, actually here, they're using this signal to actually to allow the monkey to control the robot. And they have actually several examples of this kind of thing. Okay, so that's a decoding approach. Wow, that's impressive. I realized this would be such a pain. Okay, okay, so now we come to one major reason why people have not stopped doing this. It's still a very common technique in neuroscience, but why we are sort of looking forward to new advances that allow us to measure neural activity in vivo. And that is that in many cases, it's impossible to determine what kind of neuron you're recording from. So let me give you an example. So in the cerebellum, for instance, there are Purkinje neurons, which fire at extremely high rates and very distinctive kind of output potentials. And so if you're recording from a neuron that's firing at 85 hertz or something, you know it's a Purkinje neuron. There's no question about it. But there are several kinds of interneuron in the cerebellum as well. And what if you're interested in those interneurons? And this is true in the cortex as well. You can usually determine a pyramidal neuron, but not necessarily one of several kinds of interneuron. Their frequency of action potentials looks very similar. The shape of the action potential, which is governed by things like how long different ionic conductances are open, are very similar. How do you tell whether it's one kind of interneuron versus another? This is a real problem. With electrophysiology, you can't do it. The only methods that I know about that allow you to do that are either intracellular methods, where you can fill the cell with dye, or the juxtacellular method, where you can label a neuron, and then go back and reconstruct and then stain it and ask what kind of antibodies it binds, what kind of receptors, blah, blah, blah. Well, I mean, if you're going to do this, I mean, think about the yield of neurons, right? So Miguel is recording from 1,000 neurons at a time, while we're going back and reconstructing with our juxtacellular method 1 neuron at a time. I mean, this is crazy, right? So that's why the ideal way of doing this would be is if you were able to express some kind of indicator of these neurons that was, say it was colored, say it was fluorescent. Every single time the neuron fires an action potential, fluoresces a little bit, so that with the right optics, you could determine when that neuron fires action potentials. That is the one of the sort of latest holy grails in neuroscience methods. And we're not there yet, because the method that we have now to do this is calcium imaging. So detecting when a change or an increase in calcium concentration inside the cell occurs, which is an indirect method. There are not voltage indicators yet that are sensitive enough and fast enough to actually reflect action potential changes. But every time a neuron fires action potentials, the chances are that calcium channels are also going to open, and there'll be an elevation in intracellular calcium. And so if you have an indicator of calcium, then you can determine when the cell is active. And these methods now have started to take systems neuroscience by storm. So to introduce them, let me tell you about all the different ways that calcium can change in the cell. And this is by way of saying that maybe as good as these methods are and as exciting as they are, we still are not 100% certain where the calcium actually comes from. So in this diagram, it shows all the different possible sources of calcium in the cytosol. Okay? So let me use my trusty pen here and circle them for you. Okay. So one example is here, the endoplasmic reticulum. It's full of calcium. There are receptors on the endoplasmic reticulum called ryanodine receptors. Various different things bind to them and cause the release of calcium from internal stores. The internal stores are in the ER. Okay? Another possibility is, let's see, there are various different kinds of channels, such as nicotinic acetylcholine receptor channels, NMDA receptor channels, and certain AMPA receptors. All of these pass calcium. Okay? And then there's various other things, like if you, certain metabotropic glutamate receptors, when they bind, when glutamate, they will ultimately cause, through a second messenger cascade, release of calcium from intracellular stores. So there are many ways to elevate calcium in the cytosol, but the one that is perhaps of most interest is this one here, which is voltage-gated calcium channels, of which there are a whole bunch of different varieties, and most neurons express them, if not all neurons. And the thinking goes that a rapid change in calcium is most likely going to be due to a calcium channel activation, which is going to be the result of depolarization up to the level of spiking, more likely than any of these other methods. Although this is a big caveat, as you can see here. You can imagine a highly depolarized neuron that is getting a lot of glutamate, might get a lot of NMDA receptor activation, and about 6 or 7% of that current is calcium. So it's not necessarily the case that it's coming, all the calcium is coming from voltage-gated calcium channels if the cell is depolarized. So how does this work? How do calcium indicators work? So the idea was originally, before we had genetically encoded calcium indicators, which I'll talk about in a minute, there were just these dyes, okay? So one of these dyes is called Fura2. This was used commonly in neuroscience for many, many years. So this is a structure of Fura2. the idea is that this is a fluorescent molecule. So if you illuminate it with 350 nanometer wavelength light, it'll fluoresce at about 505 to 520 nanometers, okay? The amount of fluorescence will actually decrease if this fluoro2 binds calcium. So if calcium is present, fluoro2 will bind it, and the fluorescence signal will decrease, okay? So this is one of the first calcium indicators. It's pretty much useless as a substitute for electrophysiology, right? Because it doesn't solve the problem, right, of which neuron is which, right? But it is useful if you want to see the level of calcium in the cell, and this is great. So this was used in neuroscience for many, many years. And then based on this idea that the fluorescence can change based on calcium binding, people invented these, GECIs, genetically encoded calcium indicators, of which there's now several, maybe 2 or 3 different varieties. And the most popular one is called GCAM. So this is the structure of GCAM. It's based on a green fluorescent protein molecule, okay? They stuck this calmodulin onto it, and this sequence called M13, which I actually don't understand what that actually stands for. But the idea is that the calcium will bind the calmodulin. That's what calmodulin is very good at, binding calcium. And that causes a conformational change, so that now the M13 is closer to the calmodulin. So green fluorescent protein, as the name implies, is fluorescent. If you hit it with 485 nanometer light, it'll fluoresce. But this particular variant will fluoresce a lot more at 515 nanometers if you hit it with this wavelength of light, if calcium is bind and the system here is in this conformation, okay? So it's that simple. The greater the calcium, the more is bound to this, this genetically encoded indicator, and the greater the fluorescence signal at when you illuminate with 485 nanometer light, okay? So what does it mean that it's genetically encoded? It means that you can package it into a virus, you can infect neurons with it, they will express this, and you can through certain molecular wizardry, you can have this expressed only in certain neurons. So for instance, by having a promoter that drives expression in only certain kinds of cells, you can limit the expression to only some kinds of cells. There's various other techniques that I won't get into for limiting expression to particular cell types. For instance, using Krelok's recombination. That would be a whole other lecture to explain that, which I'm not going to get into. So one of the nice things about GCAM is that the intensity of fluorescence can be normalized for the concentration because there is a wavelength called the isobestic wavelength, which if we needed another jargon term in neuroscience, I didn't realize it, but now we have one. At the isobestic wavelength, which for this molecule is 405 nanometers, if you hit this with 405 nanometers of wavelength of light, it will fluoresce, but the degree of fluorescence will be the same whether or not calcium is bound, okay? So this is really nice because then, of course, if you have a genetically encoded indicator and some neurons express a lot of it and some neurons express a little, well, some neurons are going to be brighter than others. How do you know, how can you then compare across neurons, for instance? Well, you can't really, but you can use this isobestic wavelength and basically normalize the fluorescence to that. Okay? So it's very useful to have this isobestic wavelength. The other thing it allows you to do is that if there are changes in fluorescence due to artifact, let's say your fiber bends a little bit and you get some weird fluorescence because of that, that'll be roughly the same at the isobestic wavelength versus the, if you're illuminating alternately with the isobestic wavelength versus the calcium exciting wavelength, okay? So a typical experiment, there's actually 2 ways of using this. One is called fiber photometry. And the basic idea behind this is that you express your GCAM into some population of neurons, and then you implant a fiber into the brain, and an optical fiber, like a fiber optic probe, and then you connect that to essentially a detector, right? And with some dichroic mirrors and whatnot, you can bombard your light, your brain, the section of brain that your probe is, with either your blue laser of 473 nanometers. So this is close enough to 485 that it will excite, and then you'll get fluorescence proportional to how much calcium is bound, or with the isobestic frequency wavelength. Okay. And so then with some sort of electronics wizardry here, you can detect the intensity of the fluorescence, and then you can determine how much calcium is arising from the neurons that you record from. So fiber photometry is kind of a bulk method, right? A typical fiber is something like 200 microns. Well, there's a bunch of 25 micron neurons below it, and you're going to collect fluorescence from all these neurons, or from some subset of them. Maybe only some of them fluoresce and some others don't, because only some of them are active. But what you pick up is just a certain degree of intensity. It's just a number. It's this much fluorescence. And so that's an advantage and a disadvantage. The bulk fluorescence signal is going to be fairly low in noise, which makes this a very easy technique to use. Once you've worked out the parameters for the electronics and so on, and you've worked out the parameters for viral expression, this is actually relatively easy to do. It can be used to image calcium in axon terminals as well, because terminals, whenever there's an action potential, there'll be calcium influx. That's the job of the terminal, after all, is to release neurotransmitter that's dependent on calcium. There'll be a different complement of calcium channels in the terminal, but the nice thing about this is that you can inject your virus to drive expression in the cell bodies, and then put the fiber optics in the terminal and only image from the terminals and not from the cell bodies where your electrode is, if you understand that, right? So you can determine how a signal arrives in a terminal region as opposed to just looking at what the cell bodies, which could actually project to multiple different regions. Okay? And this is easily performed in freely moving animals. But because it's a bulk signal, people are also interested in understanding how individual neurons respond. And there are a couple ways of doing this, which I'm not going to describe in detail, because that would involve describing both confocal microscopy and two-photon microscopy, which is way beyond the scope of this lecture. But the idea is that you can hook up these different kinds of microscopes, and you can use those to excite with your neurons with the wavelength of light that excites these cells that have the same problem as before. Let's try this. And this is what, I'm just going to show you a video that shows what it looks like. Okay, so this is, these what look like branches here are actually blood vessels. And you can see individual neurons are sort of lighting up, literally lighting up. It shows you the fluorescent signal from individual neurons. I'm just going to back this up a minute because we want to see this a little bit better here. And you can see that over time, there's different neurons are fluorescing, at different times and at different intensities. And so in this particular preparation, it's actually a fairly large field. So they have, maybe several hundred neurons. And this just shows over time the plot of the intensity of the fluorescence signal over time on the x-axis. And you can see they have many, many, many different neurons. And so this is a tremendous amount of data that we can analyze just from one experiment to get a whole bunch of different neuronal signals. Typically, the number of neurons that are recorded is a lot smaller than that. This is a very large field. But that gives you an idea. Okay. So it measures in individual cells. Especially with two-photon microscopy, this gives you very, very good spatial resolution. I'm not going to explain. This is best for the cortex because to image deeper structures, what you actually have to do is remove the brain that's on the overlying that's overlying the structure. So, for instance, if you want to image the striatum, you have to take off the cortex and put your objective right up close to the striatum. And in fact, this is best done in head-fixed animals because these microscopes are fairly large, right? But people are actually developing. This review that I took this image from was published in 2012. And in the last 6 years, there have been several developments along these lines where people have made, they're called either endoscopes or portable microscopes that allow you to do essentially two-photon microscopy with these very miniaturized systems. And the idea is ultimately to be able to do this in behaving animals. And there are a few labs that can do this, but it's still very rare because for a variety of different reasons, it's technically extremely challenging. You have to use this kind of lens, a gradient refractive index lens, which is essentially a small diameter lens that has a series of prisms in it that essentially allow the light to be focused in the right plane. So this is really under development, but I think over the next, say, 5 to 10 years, this is going to become more and more common where you can see this kind of imaging of individual neurons in deep structures in freely moving animals as opposed to head fixed with a giant microscope stuck down into the ground. Okay? All right, so those are calcium imaging methods. And I knew that by now I'd be running out of steam, which is why I'm only very briefly going to talk about neurochemical methods. These indirectly reflect neural activity by measuring, say, the product of neural activity, which in many cases is neurotransmitter release. So it essentially measures the amount of neurotransmitter or the concentration of neurotransmitter in the extracellular space. There's typically very poor spatial resolution with these methods because the probes are fairly large. And the amount of neurotransmitter can be influenced not just by the release of neurotransmitter and those processes, but also by the breakdown of neurotransmitter and by the reuptake of neurotransmitter. So there's certain disadvantages of this. On the other hand, if you're really interested in that neurotransmitter, then it makes a lot of sense to just measure that because that's like the outcome of all this neural activity is going to be the release of neurotransmitter. One of these is microdialysis. So you have a dialysis membrane in a U shape like this. You flow a fluid through it, and so artificial cerebrospinal fluid, and so any kind of small molecule that's in the brain, in the vicinity, in the extracellular space, will diffuse from high concentration to low concentration, will go inside, and then you collect the perfusate and analyze using HPLC or mass spec or what have you, to very precisely analyze the contents of the extracellular fluid, including neurotransmitters. One drawback of this is that to get an exact concentration, you have to use a technique called no-net flux microdialysis, which I'm not going to describe, but it's very time-consuming. And the spatial resolution is pretty poor because the diameter of the probe is something like 0.5 millimeter or a millimeter, and it's typically a couple of millimeters long. So you're really integrating over a very large volume of tissue. And the temporal resolution is also poor because the fastest microdialysis that I've ever seen is about 1 minute interval between collecting samples. And typically it's done in 5 or 10 minutes. So you're really measuring very slow changes. And of course those changes are the result of spillover out of the synapse into the extracellular space. And so what you're interested in is how the neurotransmitter is affecting the receptors in the synapse. This will give you a very indirect measure because it's measuring something that's happening long after the transmitter has actually activated the receptors. Another method is fast scan cyclic voltammetry. There's also various variants of these kinds of electrochemical methods, but the idea behind all of them is the same, which is that some neurotransmitter species can be oxidized, and any kind of molecule that can be oxidized that happens at a specific potential, called the oxidation potential. So this carbon fiber electrode, when you apply voltage to it at, and I should say, what's the voltage for dopamine, the oxidation potential? 0.7? Yeah, I thought it was 0.7 volts. So what they've done here is that they're ramping up the voltage upwards and downwards, starting at minus 0.4 volts up to 1.3 and then down again. And so at 0.7 volts or whatever it is, that's the oxidation potential for dopamine. It becomes oxidized dopamine orthoquinone. When this happens, when you apply that potential, of course, a current is going to be generated in proportion to how much dopamine is there, and then you will measure a signal based on that. And then, of course, the dopamine orthoquinone can be reduced back to dopamine, although that's going to happen at a much smaller current is going to be generated because there's not going to be very much dopamine orthoquinone in the brain after you've just oxidized some of the dopamine. And so over time, the voltammogram looks like this. The color is basically the current that happens. This is the time in relation to a certain event that happened. In this case, we presented a conditioned stimulus. Here's the oxidation potential. You can see we got this nice hot spot for dopamine. And the reason for using this kind of silly map here, color map, is because you can get changes in potential, changes in current at different potentials at various different times in relation to an event. And you want to be able to exclude those. So that just summarizes a neurochemical method. And so just to sort of quickly describe the advantages and disadvantages. Again, this provides somewhat better temporal resolution, something like 100 milliseconds than microdialysis. But in fact, if you look at, say, a dopamine signal, it can last for several seconds after a stimulus. But in fact, the dopamine is probably only active within the synapse for a few tens of milliseconds or less than that. And so again, this is a very indirect method because the changes last for so long. They're basically being filtered by the fact that there's diffusion out of the synapse. And what you're measuring is, with this method as well as with microdialysis, is this sort of filtered signal of a neurotransmitter that's out of the synapse, whereas what's really important potentially is the actions of the neurotransmitter within the synapse. There's only some neurotransmitters that can be measured with this. And yeah, so some neurotransmitters are better than others. There's very, very strong reuptake for certain neurotransmitters. For instance, glutamate has extremely strong reuptake. So to measure a prolonged extracellular signal would be kind of strange because glutamate is toxic, and that's why reuptake is so strong. And so you can't really use this method for all neurotransmitters. Okay. So like I said, this was going to be very brief with neurochemical methods. On next week, we're going to have, rather than me lecturing you, we're going to have a paper discussion. I asked Claudio, can you email the papers, the link to the papers that we're going to discuss? And the idea is that what I'd like you all to do is to divide into two groups and decide how you're going to present two different papers. Those are the two different papers. These are both papers on reward prediction error and dopamine, and they use different methods to measure neural activity related to prediction error. One of these papers, the Hallerman and Schultz paper, has a very, very short method section. So this paper over here has an expanded method section that describes in more detail how this kind of experiment is done by that lab. And that's just a short, couple of paragraphs on the actual method. If you aren't familiar with prediction error, this review is fairly short, and it describes a lot of even current thinking about prediction error, even though it's 20 years old. And the whole idea behind the journal club presentation is, like any journal club, you want to describe the paper, why they did the experiment, what they did, what the results were. You want to describe the author's interpretation and then evaluate it. Do you believe the results? And then, because this is a methods class and a discussion of methods, the real important question is, why did they use the methods they used? What other methods could they have used? And what methods are also nowadays available to answer similar questions? Those are things to be sort of thinking about as you read these papers. The papers are actually not that difficult. The Howe and Dombeck paper is a little bit longer and more involved. But it's also a kind of a very nice paper and a very nice illustration of current calcium imaging methods. It's a very recent paper. Okay? So, any questions? I was completely clear the entire time. Okay.
Se vocês quiserem perguntar em português, a gente traduz. Disse a Cani. Pois é.
Yeah, oh yeah, sorry.
I would like to know about your opinion for single unit recording. In your opinion, what are the more important cares that we need to have for the recording, like reading internationals? Like the grounding, like the connections, or
right. So I mean, the basics are to have obviously a low noise recording, right? So you can have noise as you know from the lights, from that kind of thing, from 60 cycle from the current, because there's a potential from that. If your electrodes are picking that up, then it's a disaster, right? But assuming you have all those things worked out and you have a low noise signal, then probably the most important thing is to be able to separate one unit from another one, right? So with the kinds of low impedance electrodes that I know that are being used in your lab, which are very similar to what we use in my lab, it's really a big issue to be able to separate. You might have two different neurons producing a similar waveform, and if you only have one electrode, the way you do that, I alluded to this but didn't describe it. The way you do that, and the only really way we have, is you rely on the fact that neurons have a refractory period. So once they fire, it's a few milliseconds before they can fire again. And so what you do is you take you construct an autocorrelogram. So you figure out when the neuron fires and you ask when's the next time that it fires. And if that happens at very, very short latency, say a neuron fires now and then 2 milliseconds later it fires again, that can't be the same neuron, because in most tissue, neurons can't fire at 500 hertz, even twice, right? So it's probably 2 different neurons. And so then you have to worry about that and maybe throw that out. Now, I know in your lab you use stereotrodes, which allow you to do something similar to what the tetrodes are, and that really reduces this problem a lot, but you still have to be a little bit concerned about it, because potentially a distant neuron might be sort of equidistant to both electrodes, and it's still possible, So that's probably the biggest thing that you need to worry about with this is to get good data. You have to make sure that when you say it's one neuron, it really is one neuron, and you have to be fairly confident about that. Otherwise, you can kind of screw yourself up. So. Yes.
Dúvidas então, pessoal?
Professor, I'm not familiarized with electrophysiology, but you can merge both LFP and spikes in the same study. And what kind of example could be?
You mean, so how would you use that kind of information? Yeah. So let's see. So because LFPs give you information about the synaptic potential, you could imagine that you have a lot of neurons that fire, action potentials, but not all of them, right? Or you're recording from 1 or 2 neurons, but you see a very large LFP signal at the same time. The LFP actually might be more reliable than just recording from a couple of neurons, in the sense that if you have, so action potentials are all or none, but sometimes they're none, even though the neuron is being depolarized, and all the neurons around them are depolarized as well. So if things are really synchronized well, right, and you have a lot of depolarization at a lot of different synapses nearby, the LFP will actually give you a much better indication of the synaptic input that's coming in than the fact that you're recording from one neuron that occasionally fires an action potential, right? So sometimes, to do it simultaneously, you can get information from both of these things and analyze them in sort of a similar way to understand what's going on. So that's one example. Another that's often done is to look at oscillations. So for instance, if you want to know when, say, the theta rhythm is active, you might be able to use the same electrode that you're recording action potentials with to pick up the theta rhythm, say in the hippocampus or wherever you happen to be recording from, and that can tell you something about what the animal's doing and whether he's paying attention and so on, because these rhythms are sometimes correlated with things like that. And so typically I think the way this is done is you actually have other electrodes that are optimized for LFP, but you can, in theory, you can do it using the same electrode.
I would like to thank you again for the pleasure class. It's very interesting. So we'll have this class recorded. So we will allow you to